Welcome to Faculty Focus, the podcast of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences of the University of Antwerp, live from our annual Faculty Research Day, where we highlight the research of our professors and PhD researchers. I am Teun.
And I am Marleen.
Next on our Faculty Focus podcast table, we have PhD researcher Paula Polanco Miquel. She works at the Microglia and Inflammation in Neurological Disorders Lab at the VIB Center for Molecular Neurology. Her research presentation today is titled Lysosomal Failure Triggers Microglial Degeneration and Accelerates Neurodegenerative Pathology. Hi, Paula.
Hello. It's not an easy title.
No, it's not an easy title. And that's not an easy subject as well. But before we take a closer look at your research, can you tell us what those microglia are? What do they do exactly?
Sure. So these cells are the sort of like the guardians of your brain, and you can understand them as the immune system of the brain. So basically, they take care that everything that is going on in the brain, it's flowing smoothly. So they are, for example, very important in any sort of disease context.
Okay. And so that's why they are important, I can imagine.
Exactly.
Okay.
Yeah.
Okay.
Especially like in the context of neurodegenerative disorders, where there is like a disease ongoing. These are the cells that will take care of the neurons and that everything that is going on around.
Okay.
And they also rely heavily on their endolysomal system.
Exactly.
Yeah, this is also called the recycling center of a cell. Why do we need cleaners in our body?
So first off, indeed, we have this organelle within the cell that is called the lysosome. And as you said, the lysosome basically, you can understand it as the stomach of the cell. So in many pathological conditions, there needs to be a lot of clearance of the surroundings, and especially in neurodegeneration, because there is neuronal and cell death occurring, these cells need to be cleared up. And apart from that, in these diseases, there is also protein accumulation, sometimes extracellularly, sometimes within the cell. So the protein that is accumulated also needs to be cleared up. And basically, the microglia cells try to help with this. Doesn't always go well, but...
Is there also something as too much clearance? Too much clearance?
Yeah. Well, so this is like a discussion currently in the field, because indeed, on one hand, you want the microglia to be okay and like helping and cleaning up among the many other functions that they have, but talking specifically about this. But then there's also the discussion that sometimes these cells can get overactivated, we say, and these cells can enter like this feedback loop where it can become detrimental. So they can become too activated and maybe even phagocytose. So that basically means like engulfing something. Like it can signal for phagocytosing cells that should not be like engulfed, for example. So that's kind of what like my perspective is, that this needs to be really tightly regulated. So we need to be careful on how much we push these cells for like their activity.
Right. And an important lysosomal protein is progranulin.
Yeah.
Yeah. Where does it come from, and what does it do?
Yeah. So progranulin is it's a lysosomal protein. So indeed, it plays a key role within the lysosomes, and it is interesting because there are more than 70 loss of function mutations that have been identified within this gene, which basically leads to reduction of the protein. Sometimes it's 50%, sometimes it's 100%, sometimes it's even like very small deletions that have predicted loss of function. The general notion is that it has inflammatory activity. So basically, it's understood as a modulator of inflammation. And it's interesting to study this within microglia because, as I said, they are the immune system of the brain. And when you look at the expression levels of progranulin within the brain, microglia is the cell type that expresses progranulin the most. So that's why I put those two things together.
Okay. And what happens if progranulin doesn't function properly?
So what the most, let's say, common characteristic is lysosomal abnormalities. Especially what I have seen particularly is that this leads to problems in the degradative system of the cell. So basically it will, on one hand, for microglia, it will activate them a little bit more. These cells seem to be having more phagocytic activity, but the problem is that they eat up more, but then they cannot degrade it. So that's the feedback loop that can push the cells towards cell death.
They stay full.
Yeah, exactly. It's like they cannot degrade it. And then what I particularly saw is that because they cannot cope with this, then they will degenerate as well. I think in neurodegeneration, for a very long time, the field has focused a lot on the neurons, and in neurons dying, but nobody really questioned, okay, what about the other cells? Do these cells also die? So that's why we took it towards this side.
And for your research, you use a xenograft model?
Yeah.
Can you explain this to a non-scientist like me?
Yeah, basically, xenotransplantation or xenografting means taking cells, tissue or organs from one species and putting it into other species. And in my case, we xenotransplant human microglia into the mouse brain to look at how they behave.
Okay. There is a link with amyloid beta plaques that are one of the main hallmarks of Alzheimer's disease. Can you explain a little bit how that is linked?
Yeah. Maybe I can start by saying progranulin is mainly associated with frontotemporal dementia. It's another type of dementia that leads more to behavioral abnormalities, like changes in personality. But as I said, there are also small mutations that have predicted loss of function. So it won't be like a 50% reduction of the protein, but probably you lose a little bit. And this is associated with an increased risk for developing Alzheimer's disease. Now, in my research, I worked also with granulin deficiency in microglia in the context of Alzheimer's disease, because I want to study how these cells react under neuropathological conditions. And AD mouse models are a great model for this because you have A beta plaque accumulation, which is a pretty extreme model of neuropathology.
When preparing this podcast, we come across a lot of mice research. Mice have taught us a lot in science, right? Do we appreciate their contribution enough?
Right, that is a good question. Definitely, as a researcher, I can tell you we really do. Yes. And when we work with mouse models, there is a lot going on behind the scenes and a lot of regulations and a lot of certifications that we need to first acquire to make sure that we give these animals the best possible life. So indeed, they have been huge contributors to this, and especially for neurodegenerative or just the neuroscience field, the brain is a pretty difficult organ to study, right? Yeah. So in this case, I would say we use them when there's just simply no other way to look at it better,
Yep. Back to your research, that microglial fragmentation and degeneration haven't been described before in the context of neurodegeneration. Mm-hmm. What could this new finding lead to, and why is it so significant?
Well, it's because, as I was saying, the field has been focused, the neurodegenerative field has been focusing a lot in the neurons. And I think over the past decade, there has been a bit of a shift towards looking at how other cells in the brain behave and how they are. And I feel bad for the microglia, because they are the ones that are helping and that are trying to just combat these diseases. And what I saw once we started paying attention to this microglia is that they can also die, right? And I'm not saying they are more or less important than neurons. I think everything is equally important, but we need these cells to stay where they are, to be able to help the neurons.
Can they also be a part of the disease itself?
Yeah, absolutely. So I think nowadays, when you talk about neurodegeneration, you don't only talk about proteinopathy, which is like the classical hallmark of these diseases, is that there are proteins mislocalizing and accumulating in places that they should not be. But now they also, if you will ask somebody to define it, they will also describe that there is like an inflammatory response going on, which is the microglia basically responding to these insults.
And there are also solutions to restore the function of microglia. For instance, the pharmacological reacidification of lysosomes. What do we have to expect from that?
Yeah, I mean, because of what I see, particularly, I know I work with progranulin deficiency microglia, right? But at the end, what I'm looking at is how does microglia that has defects in their lysosomal system work, right? And what I saw is that the lysosomes of these progranulin deficient microglia, they are deacidified. And it's the same as your stomach. You do need an acidic environment to be able to degrade things. So that's why I used an acidifier to reacidify the lysosomes and look if we can revert these issues with degradation that we see. So indeed, when I tested those, I did see this phenotypic reversion where the blockage in the flux of the degradation was restored back to normal. And I think this is important. Generally, it can potentially be important to many other neurodegenerative diseases where you have a context where you have to really rely on this microglia effectively degrading the material. Let's also think about how the microglia are doing, you know?
You're really quite affectionate for the microglia.
Yeah, I watch them so much in videos. You know, I had to do so many quantifications that I became attached to them emotionally.
Like it,
but I guess it will be, yes, what about us, you know, who thinks about our well-being?
Okay, maybe to finish, what advice would you give to a starting PhD researcher?
I will say be resilient. Be resilient.
Like the microglia.
Like the microglia. Yeah. Just be like the microglia. Because there's going to be challenges and it's impossible not to take it on a personal level, because if you're doing a PhD, you put your whole heart into it. You will build up resilience through time, but I think it's already good if you have this mentality, this thick skin.
And find something that can help you reacidify yourself, right?
Exactly, reacidify yourself.
Yeah.
We're going to make some stickers, bumper stickers. Yeah. Okay, thank you very much and good luck with your presentation this afternoon.
Thank you.
Thank you. Lovely to have you here.
Thanks.
Bye.